Hi, I'm Elisa, and we are continuing on with the nervous system. So now we're going to talk about the cell biology of the neurons and the glia. So we already talked about the three functional classes of neurons sitting in the three divisions of the nervous system. So there's the central nervous system, that consists of the brain, spinal cord, and then there's the peripheral nervous system itself, which is a lot bigger and has a lot more reach. And so they mainly fall into a couple of categories, such as, let's just erase, there we go. The sensory or the efferent neurons, so they're the ones that are taking information in from the outside world and the inside world. I'm hungry, this is hot, that's cold, and bringing it to the central nervous system. And then, of course, the efferent or the output, the motor neurons, the ones that are going to have an effect, like cause, right? Oh, I was cold. Effect, I put on a blanket. So the structure of a neuron. So let's start with the idea of the soma. The soma, whenever you hear that, think of it as a body, right? So that's the cell body. And so when we say neurosoma, we're talking about the cell body of a neuron. And it has a centrally located nucleus, usually pretty easy to spot because it's a large cell for the most part, except in humans, a lot smaller than, say, many other animals. But anyway, it has a very large nucleus and a nucleolus. And that nucleolus is going to be important for processing lots of stuff we're going to come across as we continue with the chapters. So the cytoplasm is going to have in it the organelles, things like mitochondria for energy, lysosomes for digesting up maybe unused or dangerous biochemicals, the Golgi complex for making complicated biochemicals, and then, of course, the rough ER and smooth ER, which are going to process and prepackage some of the protein that's going to be freshly made from that mRNA. Inclusions are things we can see. So that's usually things like pigments, and it also means nutrients as well. So that's within the cell. So we can see glycogen, droplets of fat known as lipids, and also just not as common, but also melanin, which is a pigmentation. We also have the cytoskeleton, which is both the skeleton and the muscles of a cell. So they do the motion and the support. So the microtubules, as well as the neurofibrils. So neuro meaning this is a specific type of actin filament that we find in neurons. Okay. One thing we won't find is we won't find centrioles. And the reason why is most neurons are really not going through mitosis. Definitely not after certain life stages. Now, there has been some evidence to show that there can be some mitosis in neurons, but it's relatively new stuff. But generally, you wouldn't consider it as mitotically active as, say, the surface of your skin. So a neuronal process. So a process is anything that sticks out. So if you are, want to think about your major processes, think about your arms and your legs sticking out, right? So those are the parts of your body that are sticking out. And so that would technically be processes. So the two that we're going to look at are going to be the axon here. So this is the big humdinger, and also the dendrites. So the dendrites, all of these processes start off kind of dendritic or dendrites. And so what these are the branches, as you can expect, that are coming off of the soma, or remember, the body of the cell. And so most of the time they're receiving information from other cells, right? So they're postsynaptic. So other cells are saying, hey, I have some information from you. And these little dendrites, that are going back to the cell body, are going to be where they hook up and receive the information. So the next one is going to be the axon itself. So the axon is a larger nerve fiber, right? And so what this is, these are going to be neurons that are all wrapped up with connective tissue. These help us get that fast conduction all the way to super far parts of our body. Like if you think about scratching your head, and then you scratch your head, that had to go pretty far, all the way down your arm, right? Pretty intricate signaling. So an axon is how it's done. So these are larger, usually farther reaching processes. And at the end, we'll talk about that, the termini, or the part where they interact with a new neuron, is also pretty important stuff. So typically we're only ever going to see one axon per neuron. Some neurons don't even have any at all. They originate at what we call the axon hillock, and so there's this big, like, kind of trunk that connects to the soma of the cell and gets it going. So axon structure, anytime you see axo, think axon, right? So axoplasm is the cytoplasm you would find in the axon. So anytime we see lemma, think of a membrane. So like the sarcolemma is something you would find in muscle, the axolemma would be something you would find on a neuron. And then, of course, collaterals. So these are the branches of an axon, right? So if we have an here's our cell, we're sending information down the axon hillock all the way through the axon. So we're going to have little branches when we get down there, and that sometimes can attach to multiple points, so different points on a muscle or even different cells, right? Different entire, there they are, right? Terminal arborization, so that's kind of interesting. So terminal is always going to mean end, right? But arborization, like Arbor Day, it's trees. And so what you're thinking of there it is, branches, right? So these are a complex of branches at that distal end. So that's terminal arborization, how it branches out the end. The axon terminals themselves are swellings that are going to form points or synapses, right? Which is going to be an interesting concept with other cells. So they contain synaptic vesicles that are full of neurotransmitters. The myelin sheath itself is insulating layer, somewhat protective, but also helps change the pace of signaling down the axon. So this is formed by the glial cell, which we'll look at in a little bit. It wraps itself around the axon, and sometimes many of them will wrap themselves around the axon. Sorry guys, it's actually really late where we are. All right, electrical signals travel very fast down myelinated axons. All right, so the more myelin you have, the faster that electrical signaling is going to happen. So this is the general structure of the neuron that looks a little bit nicer. As you can see, here would be the soma right here, or the neurosoma. And up here is a bunch of dendrites. Okay, here's that axon hillock that we talked about, and that's giving way to this axon here. We have an axon collateral going off to this side. Maybe it's going to go talk to some other cell, Hi, right there, ready to talk, right? But also other places that this might be going, this axon, might be reaching out to a couple of muscle cells along the way. As you can see, it's got these little, like, in this case, tiny little repeats of something. They look like hot dogs on a chain. So that's where it's myelinated. All right. What else? Oh, yes, definitely. So the nucleus is right up in here, nucleolus. We could refer this also as like, let's see. Oh, and these are some of the inclusion bodies, right? So you can see they're a little bit darker. Glycogen, lipids, for example. All right, just general structure. Structural classification of neurons. Guess what? They're going to have some different shapes. So if you look here, you're going to see some that have a whole lot of dendrites on one end and maybe like one axon. You'll have others that are more concentrated at both ends, right? Ones that look like they're doing the splits. So all kinds of different types of neuron shapes, right? So let's talk about a few of those real quick, and then we'll revisit this image. Hold on, gotta get that all cleaned up. I gotta remake those in like a minute, so it makes it nice and clean. Okay, so some of them are multipolar. So we have one axon, but we have lots of dendrites. So that's most of what you're going to see in the central nervous system. A bipolar, bi meaning two, is going to have one axon and one dendrite. So this is a lot more rare. You're only going to see this in olfactory cells like in your nose, for example, retina, so in your eyes, and also some of your inner ear. So they're not going very far. They don't have to branch out to too many places. The next one is the unipolar. So one. So this is kind of interesting because this looks like a little bit of a bobblehead kind of sticking out. So it has a single process right here that leads away from the soma itself. And so that's when we think of unipolar. It's actually because we have that single process that leaves. Now that single process again can divide afterwards, but it's that one main stem, that trunk that heads out that tells us that we're using a unipolar neuron. We're going to see these mainly in scans for the cell, organs, and also ones going to the spinal cord. So anaxonic neurons have lots of dendrites, but it doesn't have any axons. So these are also going to be found in the retina, different places in the brain, and then we've actually found these also in the adrenal glands. It's interesting. Okay, here's those pictures again. So here's those multipolar neurons, lots of dendrites up here, right? Here's their axon, right? Just one main axon for the most part. Multipolar, so here's like a cell body in the middle, and we're going in either two directions. This other one, here's that unipolar, right? So we looked at that as being like the bobblehead, right? And it's really that like middle part that is going to make it, that's the uni, right? Or the one that we were looking at there. And here's the anaxonic. So it doesn't have any true axons, but we got a lot of dendrites. So axonal transport, meaning how do we move things up and down and all around that axon? So lots of proteins and peptides, are made in the soma, right, in the cell body. And things like neurotransmitters, and, just a whole bunch of things, they still have to get all the way down to the terminal point of the axon. And that can be really far. We're talking like meters here. So axonal transport is also going to use things like ion channels to send the signals, for example. Enzymes are going to be packed in there, some other more specific signaling molecules. Axonal transport can go in either direction, and there are very special names for that. So, I'll draw a picture. Boop, boop, boop. And then here's a long axon. Okay. So anterograde is going down and away from the soma, right? So anterograde. And then retrograde is going back, right? So it's returning is retrograde. Microtubules are going to help guide everything on the inside of the axon. So on the inside of the axon, there's like this network, the superhighway of microtubules, and that's how everything's going to get transported. It kind of walks up and down it like a street. The things that are walking up and down it are going to be things like kinesin and dynin. So these are motor proteins, and they specialize in either anterograde transport or bringing it back for retrograde transport. So axonal transport itself, so it can be pretty fast. Or can be pretty slow. But anyway, 200 to 400 millimeters per day, that's considered fast anterograde transport. And this is bringing things that I think are actually kind of big, like organelles, enzymes, vesicles, and other types of molecules. We can also recycle some of these materials. We're going to see a lot of that herpes complex in there. High percentage of people are infected with that, actually. It really loves to stay and move around in the neurons, knock stuff off of kinin and all the like. Also, sometimes we'll see what pathogens that result in tetany or tetanus and rabies as well. So we can see the delay in the infection or onset of symptoms because that transport time, right? Slow axonal transport, believe it or not, gets even slower. 0.2 to 0.5 millimeters a day. That's like nothing. And even that it's a stop and go type movement. And that's what's resulting in that overall lower speed. It's always going down, right? Anterograde. And it's bringing enzymes, some cytoskeleton components for reconstruction. Fresh axoplasm will be in there. So supportive cells, the neuroglia, might be considered the actual really smart parts of the cell, of your brain, for example. So we'll look into that in a minute. But for the most part, what I'll need you to do is pay attention and learn there's 6 types of cells, that they're helping neurons, and they're called glia. And then please know what they do. Okay? The next thing is that I would like you to look at the idea of the myelin sheath. Know what it is. Know what it's made of. Know that it's found around specific nerve fibers. And tell me why that's important. Next, I would like you to describe unmyelinated nerve fibers and what relationship they have to their supported cells. Also, can you talk a little bit about what we learn in terms of regeneration or wound healing in these nerve fibers. So let's start off. Supportive cells. So the glia is generally who they are. So they're the glue, right? So glia means glue. And they outnumber neurons at least 10 to 1, at least. So that means that, at least in humans, we expect there to be around 1 trillion neurons in the nervous system. So these are the cells that bring and bind neurons together. They're also the cells that rearrange them and change up the framework. Speaking of framework, they create a supportive tissue framework, cleaning, moving, maintaining these neurons, these giant neurons. In the fetus, they can guide neurons so that where they need to be originally positioned in the brain as it develops. So there's some predetermination there in the architecture of the human brain. Actually, there's quite a lot. Anyway, they cover mature neurons, helps protect them, but they stay away from those synapses so these neurons stay functional. They're careful to prevent neurons that are not supposed to touch each other from touching each other. And that helps us give very nice precision to conduction pathways, basically making your thoughts cleaner and crisper. Types of glia. There's going to be 4 main types, at least in the central nervous system. Now, the central nervous system and the peripheral nervous system are going to have their own types of glia. Generally, they're the same thing, merit off in terms of their jobs, but there's differences. We're going to look at, for central nervous system, we're going to look at the oligodendrocytes, ependymal cells, microglia, and the astrocytes. Remember we talked about the big 4. Oligodendrocytes. So those are the ones that are going to wrap themselves around and form myelin sheaths. They aid in signal conduction. They have arm-like processes that wrap around these nerve fibers and make a nice thick protein layer, covers it up, protects it, and isolates it. The next is the ependymal cells. So they're kind of interesting. So anytime you see a cuboidal cell, you know you got a secretor on your hands. And so on the apical surface, at least, there's cilia. And that means that they're kind of sweeping and cleaning something. And that something is cerebral spinal fluid. Really important stuff. So your brain is bathed in this cerebral spinal fluid all day. And the more you think, the more neurotransmitters get flushed into your brain and kind of start swimming around all the cerebral spinal fluid. So the nice thing about these ependymal cells is they come in and they help us refresh that area so that we can have clean, clear, crisp thoughts from point A to point B. Okay. The next one is the microglia. They're kind of like the patrol. They work as macrophages and they actually develop from monocytes. So they work to help clean up any damage, old cells, and divisions in the brain and central nervous system. And spinal cord. There you go. There's all of it. All right, the astrocytes. Astro for star, right? It's the star cells. So they're the most abundant glia in the central nervous system, and they're really important because what they do is they actually move these neurons and their axons into place. So they cover the brain surface, and they're the most non-synaptic regions of neurons, but they also are the ones that are creating the neural network. So they create the framework, the supportive. They also help form the blood-brain barrier. They keep a little bit of an eye on everything in terms of monitoring blood flow, but they definitely check out that metabolic need and make sure that we have all of the nutrients these neurons might need. They convert energy molecules like glucose to lactate, and they supply these or just feed them up to the neurons. So they secrete growth factors for neurons to kind of help them move in the right directions. They also are able to communicate directly to these neurons, talk to them, tell them what to do, and find out what they need. So they regulate the chemical composition of tissue fluid as well. They're constantly sweeping through it. Astrocytes or sclerosis, whenever a neuron is damaged, astrocytes are the ones that are forming this hardened scar, a tissue that fill in the space, maintain an actual physical architecture to the central nervous system. So here they are. So this would be like a neuron, for example. And then we talked about, here's a oligodendrocyte right here, right? And it's got its branches that are wrapping around that axon, protecting it, resulting in myelin. Let's see who else is here. Oh, here's our astrocytes. So you can wrapped around, little podocytes are wrapped around this little capillary here, and they're transferring nutrients and taking away waste and telling this neuron where it needs to go. Let's see here. Oh, microglia, right here's microglia, sweeping the area, making sure everything's under control. And then down here, these ependymal cells, right, making sure this cerebral spinal fluid is being produced and cleaned. So, in the peripheral nervous system, we have kind of similar, analogous cells, but not exactly. So we have Schwann cells, we have satellite cells that I need you to know about in the peripheral nervous system. Schwann cells wrap around the axon, so they're similar to the oligodendrocytes, right? Except for the Schwanns are going to be found in the peripheral nervous system. They form myelin sheaths, and that means they also are going to help in regenerating those damaged myelin fibers. The other one's an interesting idea, satellite cells, where you see a lot of satellite cells in any tissue. At least in terms of glia, the satellite cells are going to surround the somas of the neurons in the peripheral nervous system. And they're going to do it specifically in the ganglia itself. That means that they're providing insulation from those signals, targeting things, making it more precise. And also, they're going to furthermore regulate the chemical environment. Myelin. So that myelin sheath, that pretty much just lots and lots of cell fibers, is used as an insulator around an axon. It increases the action potential, which is the electric-like signal, conduction, velocity, meaning it speeds it up. It gets that message there sooner. That's a faster email now. It consists of the plasma membrane of the glial cells, and it's got a high proportion of protein and a high proportion of fat. So it's formed again by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. Myelination also begins pretty early, the 14th week of fetal development, and it proceeds really rapidly after you become an infant. The late point of adolescence is when it's supposed to be wrapped up, and dietary fat is important to central nervous system development. Humans need a lot of energy, and a lot of energy from fat. Okay, well, that's all for this particular set of information. So hopefully you will be able to find this super helpful. All right, have a lovely day.